There we go. Hi guys, I'm Sky.
And I'm Brooke.
And as just said, we studied at the Emerson lab this summer where we focused on modeling glioblastoma heterogeneity and treatment resistance in advanced cerebral organoid systems. Our goal at the lab is to better understand how and why these tumors grow, why they resist treatment, and find better ways to fight them. So what is a glioblastoma? It's often referred to as a GBM. It is the most common brain cancer in adults in the US, and it accounts for about 12 to 15% of all brain tumors. The median survival range is about 15 months with the most aggressive form of treatment. So it is a grade 4 tumor, the highest grade, meaning it's really aggressive, it's highly invasive, and it's very difficult to treat. The STOOP protocol was created in 2005. And it showed treatment options for patients suffering with GBMs, specifically chemotherapy, surgery, radiation. And unfortunately, we haven't seen any updates in the last 20 years.
So now I'm going to go over what a cerebral organoid is. So our lab grows cerebral organoids, which are brain-like 3D structures that are derived from pluripotent stem cells. We specifically use induced pluripotent stem cells, which are adult somatic cells that are basically reprogrammed back to a pluripotent state so they can become any cell type in the body. And we want our organoids to be as complex as possible and best resemble the human brain. So we want them to include a lot of realistic cell types, including microglia, astrocytes, and neurons, but there are many more. And here on the right, we have a photo of 2 cerebral organoids that our lab has grown. These are day 153 organoids, but they are still pretty small. And our lab uses 2 protocols that have been developed to grow these organoids. The first ever protocol was made in 2013 by Lancaster, and we currently use their 2021 protocol, along with Ormel's 2018 protocol that includes microglia. So it's a little bit more complex. And I think it's also important to note that cerebral organoids are part of a larger field of organoids, including liver, stomach, kidney, pretty much any organ that you want to study more, you can create an organoid for, and we use cerebral organoids.
So our overall goal at the lab is to use these cerebral organoids as a sort of patient avatar to help us study glioblastoma tumor progression and investigate potential treatment strategies for faster translation to patient treatment. A really good way to think of this process is sort of like gardening. So the cerebral organoid acts as a soil, and the glioblastoma pulled from the patient acts as a seed. And similar to when you're gardening, you do have to enrich the soil. Or in this case, the cerebral organoid. We want the most optimized environment for the tumor, so that way we can make sure it's generating properly. And that's why we try out these different protocols. Regardless of the protocol, though, we do have to validate the cell types within the organoids to ensure that the soil essentially is properly enriched for the tumor to be implanted. And that's why we stain and image the organoids for different cell types throughout their development.
So now I'm going to give an overview of how we grow these cerebral organoids in our lab using photos that we have taken throughout this process. And the overall length of growing a cerebral organoid can take about 120 days to reach full complexity. Sky and I were only here for 8 weeks, so we only saw about 1/3 of this process. But we started with cell expansion. So we took those induced pluripotent stem cells that I mentioned earlier and let them divide and multiply in a dish until we had enough. And once we did have enough, we were able to seed for embryoid bodies. So we took about 2,000 cells per organoid and put it in its own individual well and let the embryoid body form the 3 germ layers, the ectoderm, mesoderm, and endoderm. And then we fed these organoids neural induction media, which promotes the growth of the neuroectoderm, which is this outer ring. It's a little lighter colored, and that is the tissue that will eventually become the brain matter. And then we just continue to let the organoids grow and embed them in a matrigel droplet. And the matrigel kind of acts as a artificial extracellular matrix for the organoid. And this lets it grow in a 3D fashion instead of a 2D fashion. So we see these neuroepithelial buds protruding from the organoid. And this again is just the cells becoming more complex and differentiating into the cells that we would see in a brain. And then through neural differentiation, we just allowed the organoid to continue to grow. We can see these neural rosettes forming, which are these, it's kind of hard to see, but these loop-like structures all around the edges. And that is the formation of the early neural tube. And so the cerebral organoids are just continuing to grow and become more complex. So once you have a fully grown cerebral organoid, you want to validate that the cell types that it contains are similar to what you would want to see in a human brain, because we want our organoid to be as realistic as possible. So we do that using immunohistochemistry or immunofluorescence. And in this case, I stained for neural progenitor cells on the left using PAC6. And so the neural progenitor cells are in the pink, and the blue shows the nucleus of every other cell in the tissue using DAPI as the stain. And these neural progenitor cells are located on the inner edge of the neural rosettes, and they are in a section where there's high division and mitosis among the cells, and they will migrate outwards towards the edge and become more mature later on. But these cells are just continuing to divide to become more differentiated cells later on. And here on the right, I've stained for mature neurons using beta tubulin 3. These neurons are shown in the green, and again, the blue is the nucleus of every other cell. And as you can see, they are on the periphery of these neural rosettes, and they have become more differentiated, more mature, and are beginning to form axons and dendrites and other structures that they will be using to communicate with the cells around them.
So I did a similar staining experiment where I stained for astrocytes using the marker GFAP. The astrocytes are in the green and the nucleus is purple, if you look on those outer images. So astrocytes are a glial cell in the brain, and they act as a multitasking support crew, helping maintain environment, nutrients, etc. And it just helps us confirm in our organoids that glial differentiation has occurred, since there are types of different glial cells in the brain. And it confirms our organoid modeling is in the later stages of brain maturation. And then, okay. Thank you. Alrighty. Oh, and then this one we are just going over some of the skills we learned this summer in our 8 weeks at the Emerson lab. Quite a lot there in the 8-week period, but we learned quite a bit about tissue processing, such as harvesting, fixing, and embedding these organoids, as well as cryosectioning them. So that way we could eventually go on to stain through immunofluorescence and look at them through confocal microscopy or other imaging services. We learned about aseptic techniques, so how to keep everything sterile. 2D cell culture, 3D organoid generation, and then the photos that you're seeing there are the plates that Brooke and I were actually able to personally handle and work with. As of right now, these organoids are about 35 days old. The plate on the left is Brooke's, and the plate on the right would be mine. And those little dots are the actual organoids that we were able to grow.
So now looking forward for the future directions of this lab. So as of right now, the lab is primarily focused on solidifying a protocol to grow the most complex cerebral organoids possible, because we want them to best recapitulate the real brain environment. And then we want to develop those so later on we can engraft intact glioblastoma tumor samples into the organoids, and then use that as a model to test current treatment strategies like chemotherapy, radiation, and immunotherapies, to translate that back to the patients for a better standard of care.
Awesome. And then just last slide, of course, huge thank you to everybody that was part of our journey. Huge thank you especially to our mentors. You guys have been amazing in the lab to work with, and we feel so lucky to have had such a wonderful team.
Yes, thank you so much. This has been a really incredible experience, and we'll remember it for a long time.
Yeah. Thanks, guys.